All right. Hello, everyone. Thank you so much for being here this evening. I am Dr. Diana Lee. I'm the Associate Director of Education and Training Initiatives here at Columbia University's Zuckerman Institute. And it's great to see you all here. We are in the Kavli Auditorium at the 9th floor of the Zuckerman Institute in the Jerome L. Green Science Center. And even better than this is I know there are a lot of you tuning in online live right now. And I'm really grateful that you're spending your time with us this evening because we are here for the second lecture of the Stavros Niarchos Foundation Brain Insight Lecture Series. So if your month has been anything like mine, then I think you'll empathize with the delight that I'm feeling seeing so many gathered together in a shared interest to pursue knowledge together and to just see how enthusiastic this showing is for this topic we have planned for you. And tonight, we have a fun and inspiring evening planned all about the lives and minds of animals. And this notion of coming together to learn and discover knowledge really wonderfully mirrors the Zuckerman Institute itself, because we bring together scholars, practitioners across numerous disciplines and departments to unlock secrets of the brain and mind to better understand not just ourselves, but our entire world around us. So with generous support from the Stavros Niarchos Foundation, Columbia's Zuckerman Institute continues to foster the exceptional research and outstanding programs that we are really proud to highlight, that we're really proud that we have to highlight the way science shows up in our daily lives. So our Stavros Niarchos Foundation Brain Insight Lecture Series spotlights world-class researchers at the top of their field addressing what I believe is really fascinating, thought-provoking, scientific issues that do something really special, which is touch our day-to-day. And we hope tonight's event will give you a unique view into exactly that and leave you inspired by research happening at Columbia, across our campus, at Barnard College, and at the Zuckerman Institute as well. Many of you in the audience may also already know that this lecture series is paired closely with another one of the Institute's programs, and that's the Stavros Niarchos Foundation Teacher Scholar Program. And we're thrilled to have our cohort of 24 secondary school teachers from across New York City in our audience in person tonight. And what they do is they meet before and after the lectures and come up with new creative ways to bring brain science to life in their classrooms based on the work shared by our amazing speakers. So thank you, teachers, to our teachers and to all teachers tuning in tonight for your dedication to inspiring and training the next generation of scientists. And I would also like to thank the Stavros Niarchos Foundation and the foundation members who join us here tonight for your continued partnership and resounding commitment to helping us make brain science accessible to all. So tonight's topic is something I think many of us probably have a lot of inherent curiosity about. It's animal behavior. Some of you may have pets in your home. Some of you may be like me and have loved squids since 11th grade and pursued a PhD in it. Um, Others might have grown to appreciate New York City's wildlife, above ground, underground, in the subways. There's so many ways to connect with animals. And although these animals, they cannot really talk to us in the way that humans talk, scientists have been able to peek into their minds through careful observation and really cleverly designed experiments. What is it like to experience the world through a sense that we don't really primarily focus on, perhaps like smell? Or how about through a sense we don't even have as humans? And how does an animal's perception of its surroundings shape its sense of self? So tonight, our experts will bring perspectives from across neuroscience, cognitive science, animal behavior, and biology to shed light on questions like these. So we'll hear from two exceptional speakers, followed by a conversation between them led by Columbia University's own Dr. Jessica Zhang. She will be our host and moderator this evening. And Dr. Zhang is a cancer postdoctoral fellow in Dr. Gwyneth Card's lab at the Zuckerman Institute here at Columbia. She caught the research bug as an undergraduate studying the bumblebee and its behavior and its distribution at the Rocky Mountain Biological Laboratory in Colorado. And she's been fascinated by insect behavior ever since. During her PhD in the Department of Ecology and Evolutionary Biology at Princeton University, she studied the evolution of human specialist mosquitoes, focusing on the chemistry of human and animal odors. And now, as a postdoctoral fellow, Dr. Zhang has switched her research focus from mosquito olfaction to fly vision. So she is exploring how visual information is processed by special feature detecting neurons and their downstream partners. Throughout her scientific career, Dr. Zhang has been especially drawn to studying both the how and the why of natural animal behaviors, striving to understand their mechanistic basis as well as their ecological and evolutionary context. So for those watching online, I encourage you to use that Q&A function at the bottom of your screen to submit questions as you listen to tonight's talks. And for those attending in person, when it's time for the Q&A, Dr. Zhang will invite you to raise your hand and ask your question by speaking into a microphone so we can all hear you. So put your hands together to join me in giving a very warm welcome to our host and moderator, Dr. Jessica Zhang.
Thank you, Dr. Lee, for your kind introduction. It's my pleasure to introduce this event. And in today's event, we'll be hearing from Dr. Alexandra Horowitz and Dr. Nate Sawtelle, two experts studying animal behavior using cognitive science and neuroscience approaches. So as engrossed as we humans are in our own busy lives, it can be easy to forget that we actually share this planet with millions of other species. But today, we'll consider the question, what would it be like to actually be one of those other animals? And as someone who makes a living by studying the brains and behavior of insects, in particular mosquitoes and flies, I'm utterly fascinated by this question. Although we share the same world with these other animals, we actually experience that world in totally different ways because of our different sensory limitations and capabilities. So I'm very excited to hear from our speakers today, who will help guide us as we explore the inner worlds of other animals. First, Dr. Horowitz will help us understand the inner experience of our own pet dogs, and then Dr. Sawtelle will give us some insight into the much more alien world of electric fish. So in this event, we'll hear two 15-minute talks, one from each speaker, and then I'll moderate a discussion in which we will include questions from you, our audience. If you've already submitted a question, thank you very much. And if you're watching the live stream and wish to submit a question while the talks are in progress, again, as Dr. Lee said, please use the Q&A button to submit your questions to the panelists. Let us know particularly if you're a student or a teacher. And if you're a student, tell us what grade you're in, because we're all mentors here and we really love hearing questions from students. Now, it's my great pleasure to introduce our first speaker, Dr. Alexandra Horowitz. Dr. Horowitz is an author and professor, senior research fellow, and head of the Dog Cognition Lab at Barnard College, Columbia University. She teaches seminars in canine cognition, creative nonfiction writing, and audio storytelling. Her lab studies the behavior and mind of pet dogs, and she has long been interested in understanding the Umwelt of another animal, that is, the world as it is experienced by another animal. Her research and writing is aimed to answer the question of what it is like to be a dog. She has written 5 books, including Inside of a Dog, What Dogs See, Smell, and Know, a New York Times bestseller, and most recently, The Year of the Puppy, How Dogs Become Themselves. She earned her master's and doctoral degrees in cognitive science from the University of California, San Diego, and her bachelor's degree in philosophy from the University of Pennsylvania. Now, please join me in welcoming Dr. Horowitz to the stage.
Thank you, Dr. Zhang, and thank you, Dr. Lee, and thanks to the Zuckerman Institute for having me here, and you all for coming. It's delightful to see you all. We're starting easy with dogs before we get to electric fish. I discovered dogs in grad school. I mean, of course, I had already discovered them. I lived with them, I'd grown up with them. I knew them, right? I knew what they wanted and thought about. We all do, right? That's why they're living with us and sleeping in our beds and on our couches. But what I found out is that we don't know them, right? We think we do, but we don't really understand what's on their minds. What I discovered and what I want to tell you a little bit about today is that through close examination of their behavior and a better understanding of their perceptual abilities, we can start to draw a picture of what it's like to be a dog. If you don't believe me that you can learn about animals from their behavior and how they look, here's a good example. For most of human history, when we thought about bats, this is a little brown bat here. Let's see if I start the video. We thought they were blind. People thought they navigated in the dark by touch. It was only when looking at something about their anatomy, look at the ears on that bat. They're huge. That researchers Donald Griffin and his colleagues set up a simple naturalistic experiment. What we have here is, sorry, getting ahead of myself. Let's see that video again, is the wires that they set up in the room and set a bat in a dark room and set a bat to navigate among, and found that they never hit the wires, right? Then they did naturalistic experiments where they plugged their ears, blindfolded them, closed their mouth, and they finally figured out that they were navigating through what they then termed echolocation. They were sending out high-frequency sounds and listening for them to come back. And through experiments, they found that they could see the distance and texture of objects, that they could see the speed of moving objects, like one of Dr. Zhang's mosquitoes. Griffin and his colleagues were looking at their anatomy and looking at what they actually do in the world, and then doing naturalistic experiments to probe a little more what they perceive and therefore know. And this is basically what I want to do with dogs. So 2 steps with dogs, because they're sort of special species, because we think we know everything about them. One is breaking down what we already think we know about what dogs feel and what they want. And the second is building up what they're showing us in their body and behavior that they actually know. Okay. This is the dog we think we know, right? We treat them like little people. We anthropomorphize them. This is a natural human tendency, and it has long-standing. We've been anthropomorphizing for thousands of years, assuming that because they live with us, they are like us and even want to do things that we like dressing as a Happy Meal. And I'm of 2 minds about this, because I think anthropomorphizing makes them interesting to us, allows us to be interested in looking at them. But it also restricts our seeing who they actually are. We're quick to assume that we know what's on their mind by looking at their face, for instance. But this isn't always right. I challenge you to look at these and not see smiling, right? But the dolphin and the axolotl, those are fixed features of their faces. They're not expressing happy. That's how they always look. The dog, in this case, sometimes they're smiling when they're panting. Sometimes they're smiling when they're feeling concerned and are doing an appeasement gesture to be sure that they're not punished. The chimp, the smile is a fear grin, right? That's a sign of fear. So when we think we know what they're experiencing by how they look, we can be misled. This might seem benign, but often it's really off the mark. And this is a very common thing that any of you with dogs will recognize. These are elements of dogs showing the guilty look. In one of my studies, I show actually that this look isn't caused by guilt at doing something wrong, or at least the dog isn't showing the look when they've done something wrong. It's a reaction instead to the human who finds them and thinks that they've done something wrong. We often don't know what they're experiencing or what they're thinking, what it's like to be them, but we make that first step, that first assumption that we do. So we have to get rid of those anthropomorphisms first, and then what's the alternative? How can we know what it's like to be a dog? This idea of the Umwelt of the animal was famously asked by a philosopher named Thomas Nagel about bats once he learned about their echolocation. He thought we couldn't know what it's like to be a bat. By contrast, I think that we can take a kind of imaginative leap into what it's like to be another species if we tamp down our anthropomorphisms and instead just look carefully at their behavior and their perception. So let's do that with dogs. Let's start with their visual perception because we care so much about vision. So we can understand the visual world. We think that the world is visual, but that's just our specialty. In this case with dogs, if we look at it, they have two-color vision. So what they probably see is just a muted set of colors. They're similar to us. They're nearsighted, like some of us are. So what's in front of them isn't always very clear. And then some differences start to emerge. For one thing, we have, this is representing the eye, and the darker colors to the density of cells, of photoreceptors in the eye. We have a fovea, a lot of cells right in the center of our eye that allows the thing in front of you to be very clear. Short-nosed dogs, what we call brachycephalic dogs, like the pug, have something very similar. It's called an area centralis. More like dog-shaped dogs who have a longer nose actually have a retina that has a focus of photoreceptors that go along a horizontal band. So they're going to notice something most clearly when it's on the horizon, like suddenly a behavior of a retriever starts to make sense. They're not sitting on your lap gazing at your face. That's not clear to them, but the little thing scurrying on the horizon is really interesting to them. That's because of their visual makeup. Of course, for dogs, nose beats eyes. I would say that olfaction is their primary sensory modality, so if we want to understand their Umwelt, we have to start to understand exactly how good their nose is, and then behaviors, try to explain behaviors with that in mind. So let's talk a little bit about their anatomy to get at their olfactory Umwelt. I like to compare it to humans because, well, I wonder what we've sniffed today. Think quietly in your head of anything you've sniffed. Probably not that much, but we have perfectly good noses. We have about 5 million olfactory receptor cells. These are cells at this postage stamp sized tissue at the back of the snout. This is all window dressing here. It's all back here that the magic happens, that grabs odors out of the air and maybe converts them into a neural impulse, and we notice smell. Even with 5 million, we're not doing that much smelling. We'll get to that in a second. Dogs have hundreds of millions more olfactory receptor cells. So they have not just more, but more types of olfactory receptor cells, so they can perceive things at a lower threshold, and also more types of things. They have what I like to call a two-nose system. Really, it is this one nose you recognize, and then also, like many mammals, but not humans, a second nose. It's called a vomeronasal organ, and it's in the roof of their mouth below the nasal septum. And it's able to detect pheromones and other hormones, which all animals, including us, give off and which hold information about our health, our emotion, and potentially even our identity. They even sniff better than we do. And this is the difference, I think, this behavior that they do to use their olfactory sense that starts to really drive what's different about their Umwelt than ours. They can sniff about 7 times a second. Don't try that at home. You will hyperventilate. They have actually separate routes for breathing and sniffing. So when we sniff something in, or when you respire, when you might get in some odor into your nose, you might know if you want to get that odor out of your nose, you just exhale through your nose. Well, they have a separate route that goes to their lungs and goes to their olfactory tissues because they want to be always drawing an olfactory picture of the world. Their nostrils can work independently, so they can create a stereo olfactory picture of the world, much as we create one with our eyes. And you can see something in their sniffing that tells you about their brain. There's research that shows that when they're sniffing something familiar or enjoyable, like food, they sniff primarily with their left nostril. But when they're sniffing something aversive, like, and the stimulus that they used was veterinarian sweat, they only sniff with their right nostril. So what is this telling us about the brain? Olfaction is ipsilateral. It goes from the right side of the body to the right brain. So that's the right brain being active, and so you're seeing something about their brain with their nose activity. They also have a super stealth method of exhaling. As I said, they want to continually draw a picture of the world, so they exhale out those side slits of their nose to not interrupt the flow of air that's coming through their nostrils. And in fact, when they're exhaling near the ground, flow visualization models describe the air exhaled out the side slits as nostril air jets. And what they do is create turbulence, which is essentially sending more odors into the air for the nostrils to sniff them. If we look at their brains, this is really nice research that came out of Cornell I was lucky to be a part of, shows the difference between this area, the OP is called the olfactory peduncle, but just think of that as where the olfactory information comes in. In the human, we see the white matter connections from the olfactory peduncle are pretty minimal, very local. In the dog, they go everywhere, and importantly, I want to focus on this one. They go right here to the back of the brain. This is the visual area of the brain. So there's a direct connection between olfaction and vision, and that also is going to drive how they perceive the world. So what does this have to do with cognition? Well, in our lab, we try to use very straightforward behavioral means to get at olfactory cognition. What is it? What is their behavior telling us? What is their sniffing behavior telling us about what they know? So we've done studies on whether they can distinguish quantity by smell alone. We've asked whether they distinguish the smell of their person from a stranger. They do. And we've asked how playing scent games or nose work, where you find objects by smell, affects them. And we found that it makes them more optimistic. And one study I want to tell you a little bit more about in detail is one where we've asked if they are self-aware. Now, the classic test of self-awareness in comparative cognition is called the mirror mark test, where you present a mirror to an animal, let them grow accustomed to it, and then put a mark, literally on the chimp, it's a red mark on their forehead, give them the mirror and see how they react. The chimpanzees touch the mark. That's seen as evidence of self-recognition or even self-awareness. There's an elephant at the zoo called Happy, who has passed this test. And even dolphins, and these are cleaner fish, seem to pass this test by moving their body to see the mark where it's been applied. But dogs, do dogs pass the test? They do not. You might have a video yourself of your dog in front of a mirror. They probably treat it like a conspecific, like another dog. They bark at the dog. They look for the dog behind the mirror. They play bow in front of the mirror, or maybe they just ignore it altogether. But I think you know my punchline here. My feeling is that that's probably because this is a visual test, and they don't really care what they look like in the mirror the way we do. So we designed an olfactory mirror, and I'm sorry to say, it's not an actual mirror that reflects your smell back at you, as horrifying as that would be. Instead, what we did was we looked at their natural behavior where they leave their odor outside and investigate other dogs' odors and tried to bring that into the lab to test what would happen if the odor of themselves had a mark on it, just like in the mirror mark test. And then we compared how long they sniffed each of these canisters, and I'm just going to show you a minute of a video of presentation of a few of these dogs so you can see what it looks like to watch sniffing behavior. This is Julie Hecht, my former lab manager, just getting them interested in coming and sniffing.
Okay, free.
And really, all we're looking at here is how long they're sniffing each of the tests. Canisters, one might have their own smell, one another dog's smell, and another one will have their own smell with a mark. The dogs are all individuals. They behave quite differently. But I think you'll agree, you can see they're thinking about it. Some dogs show a disgust response. We often get that. So that just shows you a little bit what it's like to study olfactory behavior in a laboratory setting. So here I just want to show you a little bit of our results. What we found is that they sniffed other dogs' odors significantly longer than their own. This is the length of time they sniffed. And this was just reproducing in the lab what we see on walks, where they're interested in other dogs' smells. And this was the key condition for us. They smelled their own odor a certain amount, but significantly longer when it had been marked with another odor. And just to be sure that they weren't just interested in that odor, it was a really good odor, we presented their own smell marked with just the mark by itself. And we found that they were really only interested in that marked smell if it had changed their own odor. So the conclusion I'd like to draw is that that behavior might be indicating that they're recognizing their odor as themselves, that's me, and they show interest only when they've noticed that it's changed. A little bit of a rudimentary sense of themselves. Now, even to step back from olfaction, I'll say there are probably other ways to test sense of self just looking at behavior, and we've actually done another study looking at dogs' body sense in navigating and opening. As it gets smaller, we had a little window and we kept closing it more and more and more and asking the dog to navigate through. Here you see a head duck and an elbow bend and a rear elbow bend. Insofar as a sense of one's body size might be described as a representation of oneself, testing an animal's sense of their size in a changing environment might be another way to investigate if they have a sense of self and are self-aware. All right, I'll leave it there. I just want to thank all the dogs who come to our lab at Barnard, which could be your dog. We're doing a study right now, and all the people who accompany them. Thank you very much.
Thank you so much, Dr. Horowitz. Remember, for folks watching online, if you have a question, please use the Q&A button, and we'll try to answer them in our Q&A after the second talk. So it now gives me great pleasure to introduce our second speaker, Dr. Nate Sawtelle. Dr. Sawtelle received a PhD from Brown University, where he worked with Mark Baer on understanding how experience shapes the circuitry of the visual cortex in adult mice. He then did a postdoctoral fellowship at Oregon Health and Science University with Curtis Bell, where he began working on the sensory and motor systems of weakly electric fish. Dr. Sawtelle is currently a professor of neuroscience at the Zuckerman Mind-Brain Behavior Institute at Columbia University. The goal of his research is to link the properties of neural circuits to their functions using experimental, computational, and comparative approaches. In particular, the lab focuses on understanding how cerebellar circuitry in electric fish and mice generate predictions of the sensory consequences of action. Please join me in welcoming Dr. Sawtelle to the stage.
Great, thanks so much. So I'm going to tell you about electric fish today, but before I dive into that, I want to just say a few general words about why I study these animals and about the field of neuroethology, which for me is very exciting. Neuroethology is the study of the brain mechanisms underlying natural behavior of diverse species. There's two key ideas here that I want to get across. And the first is called Krog's principle, and this is named after a Danish physiologist that was writing in the early decades of the 20th century. And Krog said, for every biological problem, there's an ideal system for studying it. And this sounds very sensible, simple, but it's not necessarily the stereotype you have about science. You think about scientists all studying lab rats or lab mice, or maybe they should all be studying humans. But Krog thought that you should study the diverse animal that's best suited to answer your question. So we can take a few examples, and these are all actually taken for research in this building in the Zuckerman Institute. So this neuroethology approach is alive and well here. So imagine you were interested in the biological basis for aggression. You might do well to study this Siamese fighting fish. You may have seen this in the pet store. It's also called the betta fish And these fish have been bred for generations in Southeast Asia for their aggression. And people bet on these fights, and these fish are incredibly aggressive. And people are learning, neuroscientists are learning amazing things about the genetics of aggression from the studies of these fish. Or maybe your question is memory. You want to understand how we remember things and how memory can go wrong with aging. You might study these animals. These are chickadees, they're food caching birds. This species you can even find in Central Park. And to survive cold winters, these birds bury in the forest hundreds or even thousands of seeds, and they remember where they store those seeds, and they find them and survive the winter. So this is an amazing ability. Or you might choose, like Eric Kandel, Columbia's own Eric Kandel here, the very simplest animal that you could think of that forms memories at all. And this is the sea slug, Aplysia. So this animal remembers things, but it has a much simpler nervous system. So this is the first idea. Study diverse organisms, dive deep into their minds and their behavior, and I'll do that in a minute for electric fish. But the second principle that I want you to remember that really drives me is this idea that by comparing what we learn from all of these diverse animals, we can find unifying principles in biology. Eric Kandel found molecules and mechanisms for memory in Aplysia that also held for mice, and we think also hold for humans, and he won the Nobel Prize for this work. So this idea of finding unifying principles by studying diverse species is one of the take-home messages and one of the things that drives me to do neuroscience research. Okay, so here's my favorite, the electric fish, and we'll talk about it now. So first of all, why fish? So fish have been around for a long time, about 500 million years, and they actually make up about half of the species on the planet. And what this means is that evolution has had a long time and many chances to make remarkable things happen. And one of those things is shown here in this phylogenetic or evolutionary tree of fish. You see these red rectangles. Each of these represents the independent evolution of the ability of animals, fish in this case, to emit electrical fields. And these are called electric organ discharges. The organ that generates these fields is called an electric organ. So it's not an organ for making music, it's an organ for generating electricity. Okay, so why would a fish generate electricity? Okay, one reason you may have heard of already, so this is the South American electric eel. Let's see if this will play. Okay, so that was the shock, the 500-volt shock of the electric eel. And so the electric eel generates electricity to stun its prey and maybe to ward off predators. So electric fish have also played important roles and been known to humans for a long time, since antiquity, because you can feel these shocks. But they played an important role in the discovery of electricity. So Volta, the person who made the battery, first invented the battery in the 1790s. He called it an artificial electric organ and was inspired by this ocean torpedo ray, which is also electric. And if you were in the Victorian era, instead of coming to an event like this, you might gather at the local naturalist's house who had imported one of these torpedo rays, and you could feel this new phenomenon of electricity. If you held hands with the man here dipping his hand into the bowl with the torpedo, and you held his hand, you would feel the shock from the torpedo, even though you weren't touching the animal itself. So this was some of the first ideas of electricity and bioelectricity. So these electric fish have been actually influential. Okay, how do these things work, these electric organs? So it starts in the brain, and we're back to the electric eel here. So it starts with a thought in the brain of the electric eel. The eel might, we don't know what he's thinking, but he might be thinking, why does the scientist keep sticking his hand in my tank? And so neurons in the brain of the fish, just like neurons in your brain, communicate with tiny bits of electricity, so much less than a volt. So the signals in the brain go to the spinal cord, to motor neurons. The motor neurons themselves generate some more electricity, just like your motor neurons do, and those cause the release of a neurotransmitter, acetylcholine. The acetylcholine floods the electric organ, which is in the electric eel distributed along the length of its body here. The electric organ is made up of thousands of excitable cells. The acetylcholine makes them all discharge together, so these tiny bits of electricity get magnified into a giant volt of electricity, this 500-volt shock. And if this process sounds maybe vaguely familiar, you'd be right, because all of these steps are the exact same processes that let you move your muscles, right? The release of commands in the brain, neurons in the spinal cord, the release of the neurotransmitter acetylcholine, and muscle contraction. And in fact, the discovery of acetylcholine, which was the first neurotransmitter discovered, came from crushing up the electric organs of fish and isolating the chemical, because acetylcholine is found at its highest density in the electric organ of this torpedo. So this was a big discovery, the first neurotransmitter. And so this is, my attempt here to show diverse examples from biology highlighting a general principle. And we've learned many other things about electric organs, and they're very interesting, but I want to move on to the sensation part now. And so this was a question first asked by Darwin, actually. Darwin knew about strongly electric fish like the eels and the torpedoes, but he also, and other naturalists, knew about another kind of fish that had electric organs, but they didn't seem to work. And he called them pseudo-electric organs. And he thought they didn't work because if he stuck his hand in the water, like what we saw earlier, nothing happened. He could perceive nothing. And so this really bothered him so much that he actually wrote about it in The Origin of Species. And he said, why do these animals, and it wasn't just one, it was many species, why do they have these electric organs that don't seem to do anything? So that's the question. We go to the Boston Aquarium, you can see these electric fish, and if you dipped your hand in there, you would feel nothing. But if you had an electrical amplifier, and if you were able to put the amplifier wires in the tank and then play that through a speaker, you'd hear what we're hearing now. These are the electrical pulses of these fish. So they are emitting electricity. It's just a tiny amount of electricity that you need special equipment to hear, and Darwin didn't have this apparatus yet. Okay, and so what Darwin was missing was the concept of electroreception. Electric fish generate electricity, but in fact, many animals have the ability to sense electricity. And this is an ancient sense. So the first vertebrates on Earth, the lamprey, or the animals that came before vertebrates, have electroreceptors on their skin. They can sense weak electrical fields. Sharks, dolphin, platypus, echidna, even insects can sense electricity. It works best in the water, but not only in the water. So this is actually a widespread ability, and none of these animals can generate electricity, but they're all sensing it. So how does this work? So if you look, you see these little dots, maybe, I don't know if you can see them. There's little dots, little pimples on the shark's face. Each one of these is a canal filled with a gelatinous substance. It ends in this little cup called an ampulla, and then connected to that are dozens of receptor cells that send nerves to the brain. And what these are is essentially tiny voltmeters. They sense voltage differences across the skin of the animal, and they do so with remarkable fidelity. So this voltmeter that you could buy on Amazon can sense about a millivolt, is about as low as it can go. The shark behaviorally can sense a nanovolt. So absolutely minute amounts of electricity these animals can sense. Why would they want to do that? And the reason is that any animate thing, animals, plants in the water, because we're electric creatures, our bodies have membranes and excitable cells, we generate tiny, tiny fields when we're in the water. And so the shark senses these fields. So this is a behavioral study. The shark found the fish buried in the sand. Now, the experimenter covered it with agarose, which conducts electricity. The fish still found it. But if the fish now was buried in an insulating material like saran wrap, the shark swam right past. So the fish use these electroreceptors to find prey and also even to navigate in the Earth's magnetic field. So this is a remarkable ability. But we still haven't understood what weakly electric fish do with their receptors. That's what I want to tell you about in the final part of the talk. And there's three functions I'll tell you about. First, know that the skin of electric fish is also covered with electroreceptors. So it's an array of electroreceptors, in fact, thousands of receptors covering the skin of the fish. And there's two different types, and these receptors are actually specially tuned to detect the electric field of the fish. So this is an electric fish, it's generating a weak electrical field. These receptors are specialized for detecting the electric pulses of fish. Okay, so what would this be good for? And now this was work of a number of scientists. One of them was Carl Hopkins, and Carl went to Africa and did the experiment that Darwin would have done if Darwin had had devices to measure electricity. So he went to the Ivindo River in Gabon, and he put his electrode, he hiked up and down the streams and put his electrodes in the water and captured fish, and this is what he found. All of these different species, all of them weakly electric fish, and he measured the electrical fields, and he looked at males and females, and he found all of this diversity of electrical signals. So you see these little squiggles, they all have different shapes, and what he discovered is that the fish can tell the difference between males and females and between different species. So this is an entire language that these electric fish use to distinguish species, sex, mates, friends, enemies, defend their territories, etc. So this is a rich world that was completely unknown. Okay. Function number 2, and this was a laboratory discovery. So the fish seemed, it was kind of noticed that the fish, it was speculated that the fish may be able to use this sense not only for communicating or detecting other fish, but also detecting objects in the environment. And so you heard about bat echolocation earlier, and this discovery in the fish was inspired by that prior discovery. And what was found was that fish could be trained to detect, to discriminate between two identical objects, visually identical. I've done this experiment. I brought an object here. So two identical objects. The only difference is one or the other contains either a conducting or an insulating object. So this is, even as an electric fish scientist, it still amazes me to watch these fish, because if you look at this, you absolutely cannot tell whether there's a rod in there or not, but the fish can do it. And that's because this is a ceramic filter that's porous and conducts electricity. So it's electrically transparent. The fish can tell what's inside of there. So this is an amazing ability. So how does this work? So you can imagine each time the fish emits its electrical field, current flows through the skin of the fish. So there's current lines are shown in red here. Now what happens when the fish swims nearby an object that is an insulator? So a rock, for example. So this is the rock. The rock perturbs the flow of current, causing less current to flow straight through the rock and more to flow around it. So it creates what you could think of as an electrical image on the skin of the animal, a shadow, if you will. What about a conducting object? So another animal, a fish that's salty and conducts electricity more than the fresh water where these animals live, it would be the opposite pattern. More current would flow. So a bright spot on the skin of the fish. So using these electrical images, which you could think of as vision at a short distance, so this sense only goes about a body length away, or touch at some distance, the fish can discriminate the position of objects, their distance, their size, their shape, and even their electrical resistance and capacitance. So again, amazing senses that we don't have at all, and that are incredibly useful for these fish. Many of the rivers that these fish live in have very poor visibility. They're turbid waters, so these fish with this sense are masters of the dark. They're completely at home in complete darkness and can navigate complex environments using this sense. So that's function number 2. Now just back to the video. It may occur to you that in a social setting like this, that the fish are bombarded by a cacophony of electrical signals. So how do they make use of this sensing capacity when they're surrounded by so many electrical signals? And for many years, and it's been a topic of fascination and study, people have wondered and examined ways in which these fish avoid jamming one another. So how do they ignore the pulses of others? So in a recent study that we did, we asked the opposite question. We wondered whether it's possible that they could actually benefit from each other's pulses. And so to highlight the possibility, I'm showing you here the discharges of several fish in a group. And what you notice here is the actual emission of the electricity is extremely brief. So the pulses are less than a millisecond. It's just tiny, tiny clicks separated by silence. So it sounds like a cacophony to us, but for the fish, there's lots of space in between, and it's possible that they could actually listen separately to each of these pulses. And so I don't have time to go into all the details, but what we showed through some behavioral experiments, recordings in the brain, and computer modeling was that these fish actually are able to enhance their sensing ability based on the electrical pulses of other fish. And there's some basic physics that supports this, and we were able to model it and show that objects that are too far away for the fish to sense based on its own electrical field due to the steep fall off of electricity with distance, they're able to sense based on the pulse of another fish that's nearby. So these fish are sharing energy to perceive the world. And this is interesting because this actually happens in a lot of human artificial active sensing technology. So if you think of radar or sonar, it's often the case that multiple radar antennas are used to enhance sensing, medical imaging. So in MRI and PET scanners, multiple sensors are emitting energy, shining them on you from different directions and receiving them, and then algorithms work that out and give you a better, richer picture of the world. It's possible that dolphins do the same thing, we don't know, but we think these electric fish are doing it, and that's amazing. And as a neuroscientist, that leads me to wonder a lot about the brains of these electric fish. So what are the brains? How are the brains of these electric fish able to process all these information? What are they like? And that's what we study. I don't have time to tell you too much, but I'll just show you this nice picture that I like. So we typically think of the primate brain as the pinnacle of brain evolution. And this is a chimpanzee's brain. Your brain looks a lot like this. The green is the cerebrum or the cerebral cortex, and that part of the brain, that's the folded part that covers the entire surface of your brain. So when you look down on the brain, you see the cerebral cortex, and we think that's what makes us human. That part of the brain is our consciousness, our awareness, our intelligence. But there's another pinnacle of brain evolution. That's the fish that we study here up on the left. It's got a giant brain. It actually has a larger brain to body mass ratio than humans, so one of the biggest brains on the planet. And it's organized along completely different lines. So the thing that covers the entire brain of this fish is not the cerebrum, but it's the cerebellum. That's a part that you also have, but you can't see it in this picture because it's tucked under the giant cerebrum. The fish also has the cerebrum, but it's tucked under the cerebellum. So there's two ways to build a brain, at least, and these are two examples. So this is something we want to understand more about in the future. And so I'd like to thank you, first of all, my lab and funding sources.
So thank you very much, Dr. Sautel. I'd now like to invite both of our speakers to come and join us for the Q&A portion of this event. So I'm going to be taking questions from both our in-person audience as well as the online audience. And just to preempt any specific questions we have about dog training, unfortunately, we cannot answer any specific questions about training your dog. But if anyone here has a pet electric fish, please direct those questions to Dr. Sautel. So we're going to switch between in-person and audience questions. And when it's time for our in-person questions, please wait for someone to bring a microphone to you so that our online audience can also hear you. And I'd actually like to start our discussion with a topic that came in a few times online. So several members of our audience are interested in animal communication. And both of you have thought a lot about designing experiments that let you ask questions of animals and understand their answers. So given your experience designing these experiments, what have you learned about how to talk to animals and understand what they're saying to you?
Let's start.
Yeah.
Go ahead.
It's hard, is what I've learned so far. So the electric fish do communicate, so that the fish I study emit brief pulses, and the sequence or the interval between those pulses changes moment to moment, and that's their language. So if the fish are aggressive, they go really fast, but they can change the pulses in many different ways. And so that's long been thought to be some sort of language, and it's easy for us to measure. It's easy for us to play it back to the fish. And we have a project called ChatGP. So GP is Nathanemus petersi. So we're trying to be cute, and we call it ChatGP, and with people at Harvard who are brilliant at large language models and know how ChatGPT works. They're taking all of our data and trying to figure out what the fish say to each other. And so far, we've learned absolutely nothing. Without the context, without knowing about the natural lives of animals, what would they be talking about? You can be very clever and use lots of fancy computational methods. And maybe we'll succeed, and I'll have a report. But so far, it's very hard.
Yeah, with dogs, it's different because their behavior is showing us a lot about something familiar in their communication. They communicate through their behavior, and they read our behavior as communicative. Again, we have to, I think the difficult thing with a species like domestic dog is putting away a sort of preemptive assumptions about what a communication means, and instead letting the context and the behavior in repeated instances show us what they're meaning to communicate. But of course, they also have, they have very analog communicative systems to us insofar as they communicate vocally, right? They have lots of vocalizations. We mostly, interestingly, don't listen to those vocalizations or collapse them all into meaning one thing. A bark, right? You don't know that there are different types of barks, maybe, right? But they're definitely communicating different things in that modality. And then olfactory communication is challenging to understand. And I think one of the interesting things about looking at olfaction and seeing it as communicative, I look at it to see how dogs' behavior is affected by stimuli, olfactory stimuli, is that it exists at a different kind of time course than a vocal communication or behavioral communication, which is sort of happening in this moment. And olfactory communication might be happening over time, right? A communication is left, and the odor isn't received till a future time. So that's challenging. I don't really know how to do it, right? That's the work of exploration that we do a little bit in our lab.
Thanks. Do we have any questions from the audience? Please raise your hand and someone will bring a mic to you. Let's have this woman up here.
Hi, I have a question about the electric fish. So you were studying, you say it's a lot of weak electric fishes in West Africa in a river, right? Do you think the location, why they're in fresh water, is some kind of connected to why did they evolve to be weakly electric, like as opposed to like they would live like birds in air or in salt water? And also, why do the faces look like that? They look like they look like colibri, but we know like colibri like evolved to like go to this flower. Why is the fish face like that?
Yeah, so the second one first. So the fish that we study, that I showed the picture of, is called the elephant nose fish. It's really a chin, but that organ is first of all highly mobile. So the fish uses it to dig for invertebrates that it eats in crevices and rocks. So it's highly mobile. It's when you watch the fish swim, they're moving it all around, and it's also the focus of its electric sense. So it's densely covered with electroreceptors. So that's where the most electroreceptors are, so highly sensitive. So it's kind of like your fingertip. Just as your fingertip is covered with sensors and you use it to explore, the fish is using its chin in the same way. So in terms of freshwater, saltwater, so there's electric fish, there's fish that emit electricity in the ocean and freshwater, so it's possible in both. But it could be energetically a little easier for these freshwater fish to emit and sense electric fields.
Do we have any other questions? How about the gentleman in the back?
Thank you so much for two beautiful talks. So my question is mainly for Dr. Sawtelle, but maybe for both. I was wondering if there is a notion of individuality that you can draw from the signature of the electric field of each fish, and then what would the analog mirror test look like for that?
Yeah, so it's not completely clear, but it's definitely possible that so I showed you these shapes of, if you record the signal, you see this wave shape. And the tiny details of those wave shapes could be different between individuals. So they would differ by tiny, tiny amounts, but it could be that the fish can use those. And so that's something that people have tested a little bit, and there's kind of mixed evidence for it, but it's certainly conceivable that the fish can recognize individuals based on these purely electric signatures. And if that's the case, then you could do an analog of the mirror test. You could perturb, experimentally, you could shift the shape of the electric pulse of the fish a little bit and see if it notices. And actually, in our lab, we study in a broader sense this idea of how the fish distinguishes its own pulse from the pulses of other fish. And so we actually do this experiment. We actually know a lot about how that works in the brain. But it's a little bit different than what you asked, but related.
I'm going to take an online question right now. So someone is asking about spatial memory, especially in dogs. So they observe that dogs seem to have a good mapping of where they last left a ball or toy. And they're asking if there's evidence that dogs can plan ahead. What in general can you tell us about animals' ability to plan ahead?
Right, which is part of a sense of self, really, is thinking about oneself projected into the future or thinking about one's past actions. And it's actually been surprisingly hard to test whether dogs have this kind of episodic experience of the past and whether they're planning for the future. But behaviorally, they certainly do indicate that they're anticipating future events. They learn associations, many associations, which indicate to us that they're, when you get up from your chair and then start heading for the leash and get the keys, they've learned those associations only because they're anticipating what's going to happen in the future. So is that planning? Not necessarily, but they're thinking about it. And certainly caching species, like the chickadees that were brought up, I think you could describe what they're doing as planning. Now, what the experiential component of that is a little bit, I have to be a little bit agnostic about. I don't know that they're having the kind of propositional phrasing of their experience that we would think, where they're saying, I'm putting this seed here or I'm planting this bone here for later, right? But they certainly have the instinct to do that. They can remember that they've done it. They have good spatial awareness. Dogs also can navigate through olfaction, and they'll recover it later often. So that looks very much like planning, but for various reasons, we don't want to say that we know exactly what they're thinking.
Thanks. Any questions? I think there was a hand, this one in green here.
Hi, thank you all. I'm a teacher. And so I initially had kind of a question for each of you, but I think my question for Dr. Sautel could be for both of you in a way. It's how are humans messing this up? So we have all this tech, all this electrical sonar equipment we use. Is it, or is the distance far enough, given what you were saying towards the end, that it isn't really messing with the natural systems? And then I thought that it could also kind of address dogs, and that, we have all this tech that I play the radio for my dog or turn on the TV. Are they actually annoyed by that and the frequencies that I can't hear? And then my selfish question is, you said that scent was their most important sense. And so is my dog smelling my emotions when they act a certain way? If I feel anxious, anecdotally, I've noticed he throws up. If I'm sad, he comforts me, so is that more of a smell or a learned behavior given the way I reward that?
Do you want to go for it with the electrical?
Sure. Yeah, that's an interesting question. I mean, in these rivers, I don't know enough, I would say, about the rivers of Africa where these fish live to know if electrical pollution per se is a big factor compared to more general habitat destruction and pollution. And my guess would be that the destruction of rainforests and other things in South America and Africa in general is bad, and that the electrical pollution may not add so much to that. With sharks, and it's also true that this is a pretty short range ability. So if the fish was really annoyed by an electrical signal, it could probably get away from it pretty quickly. So the main natural source of noise in their environment is lightning strikes, but that's a lot of energy. So that the energy from most man-made things is probably limited. But people do, I think, try to use electrical deterrents for sharks. So it's kind of a little bit related.
I think to use Nate's word, noise, I think we create a lot of noise for dogs. I think about it actually even visually, the dogs have a higher flicker fusion rate than we do, which is to say that sort of one way of describing that is the sort of number of snapshots of the world they're taking that the brain then reconstructs at every second, that the brain then reconstructs into smoothly moving motion. They have a higher flicker fusion rate, so things we design for our flicker fusion rate are probably flickering for them, which would be annoying. That's a fluorescent light, for instance. We put a lot of, their auditory system is keener than ours. And we create a lot of anthropogenic sounds, which would probably be irritating to them on a daily level. I think about thrumming sounds or sort of like oscillatory sounds like a fan that's left on the floor type of things. There are there's so much olfactory pollution, it's completely messed up, right? Like we don't want a dog to smell like a dog, so we mess them up. We change their scent a lot, right? We don't let them experience each other's scent. We don't want them to smell us, even though that's partly how they recognize us. We bathe them, right? So and we usually don't let them sniff. We don't let them explore through smell. So I think there is a lot of room for improvement there. And as for the question about smelling emotions, it's something that has been studied, and actually we do know that dogs can distinguish between the smell of stress and happiness as represented by sweat captured on a t-shirt of humans. Now, what they're doing with that information, maybe people would interpret it differently, right? So a dog coming to you, if you're stressed, might be interpreted by you as comforting, but I don't know if that's the dog's intention or just they're noticing something different, which is definitely the case. And we're actually doing a study now where we're looking to see if they're recognizing the smell of stress and happiness of a conspecific of another dog.
Do we have more in-person questions? How about this person? Do you have a question? Yeah.
Thank you both for a wonderful talk. My question is, we're talking about communication, intelligence, self-awareness. How close are we to where science can finally say that sentience is ubiquitous in the animal kingdom? And then what does that mean ethically for us, especially you two, as you go forward and study these topics?
That's a big question. I think that a lot of people would argue that sentience in terms of just perceptual capacity, right, is present in all animals. But I think it is something that philosophers debate more than scientists. In fact, I don't know many scientists studying sentience per se. But that's not to say that it isn't a subject for science. But until recently, I don't think there were scientists studying or even using the word consciousness. And it wasn't so long ago that we weren't talking about emotion in non-human animals. And I doubt we're talking about emotion in fish, even though maybe one could. And so I think we're waiting for science to catch up with society's ways of using these words. But I think people who study animal sentience would argue, more philosophers again, would argue that it's ubiquitous, right? And we should take it, instead of saying that it's something that needs to be proven, we should take it as given and work from there.
I'm going to take an online question. So someone asks, if given millions of years in the evolutionary scale, could animals like dogs and fish reach the same or similar intellectual capacity as human beings? And perhaps this relates to the slide that Dr. Sautel showed. And if you can tell us a little bit about how you think about intelligence and of humans in an evolutionary context.
Yeah, I don't know. I think it all depends on what you mean by intelligence, which the things that we study in the brain of the electric fish are certainly, I think they're very intelligent. They're amazing. Imagine you're bombarded with signals and you're every millisecond distinguishing where a pulse came from, objects, reflections, all of these things that animals do in their sensory and motor abilities, I think are amazing. They're amazing in terms of their complexity and all of the brain processes that have to happen for those things to work correctly, in terms of really complicated networks of neurons in the brain. So all of those things, to me as a neuroscientist, those are the things that I think about as being, fantastic and intelligent. How those relate to our own, our own conceptions of intelligence as humans, I think that's a different question. That's outside of my area. But I think this idea of different ways to build a brain is a very interesting one, and the idea that we're just one branch, one possibility, I think is fascinating.
There are questions. How about this woman here?
Hi, I'm a PhD student in neuro over at Sinai, so thanks for having me. I kind of have a high level question, but I also just wanted to comment really quick on, I think it's awesome that service dogs can learn the scent of a symptom coming on, and I was sort of looking into that. That reminded me of the emotion stuff. I study emotion as well, so sorry. And the New York Animal Declaration on Consciousness happened recently. They had some interesting scientist, philosopher. I think they all came to the conclusion that there's at least a chance of consciousness, and that's what the declaration largely is. But my high-level question for both of you is, there's this debate in human neuro, in neuro in general, where cognition and action sort of break down, and we're not looking at this, cold cognitivist approach to the mind in general, but maybe it's more an activist where you can't really separate between perception and action. A lot of this debate with, the point that's brought up with humans is, hey, look at our vision, we move it around a lot, so there's active vision. I think when you look at these animals, it's just so much clearer that you can't really separate perception from action. I'm wondering how much in the comparative neuro world does this debate persist, or what the thoughts are.
In our work, we study, we don't make hard distinctions. We study parts of the brain where motor signals and sensory signals are combined, and we study those combinations and how they enable the animal to recognize itself, to distinguish objects. So for us, those signals, and I think many neuroscientists, in many parts of the brain, we see increasingly that these signals are all mixed up in the brain, and that there's some utility to thinking about sensory processing, and motor control is separate, but there's also lots and lots of space where those things fuse together, and where our ready-made concepts are not so useful, and we just need to look. We need to look in the brain and think about what's the job this part of the brain is doing, how are these signals combined to compute something of interest, and that the labels of sensory or motor or cognitive, even all of these labels, I think, need to be they're provisional, they're fine, but they shouldn't constrain us as we try to understand how the brain is actually doing all the amazing things that it's doing.
Yeah, and I would add too that, I am ostensibly a dog cognition researcher, but I'm studying what? I'm studying sensation and behavior. So perception and action, right? And cognition starts us, it just gives us a way to start talking about, or frame the question that we're asking. But I'm not sure that we're ever going to get a lot of answers to some of those cognitive questions, because I don't know that they're really answerable in the same way with non-human species. So then looking at behavior is where I think, and perception is where it really is.
Yeah, let's take a question from this side of the room. How about that person at the back?
Hello. I have maybe more of a basic question. Do all of the fish that produce electric fields have the ability to perceive electric fields? How are they not zapping themselves?
Right. So as far as I don't know of any examples of fish that emit that don't also sense. I think it's pretty much a rule that those that emit sense. In terms of the zapping, so for the strongly electric fish like the eels, they have layers of fat and connective tissue that insulate the key vital organs so that the current mainly flows around and out. So they do get a little shocked probably, and I think when they discharge, you can see little twitches, but they have enough fat and tissue that protects them. So they have specializations to prevent that.
So I'm just going to, close with a topic that came in online quite a bit. So a lot of people who submitted questions online are very interested in the applicability of animal work to humans. So would you say that your research has, there are direct applications of your research to, for example, human brain or psychology research, or do you instead find there are just more subtle ways in which understanding the inner lives of these animals can enrich how we think about ourselves?
I mean, with dogs, I think it's easy because one of the ways that it directly affects us is in our interaction with these creatures, right? To understand better the world they're experiencing, I think, is going to create an improved relationship with your dog if you are acknowledging what they're saying because you understand how to interpret their behavior. If you're acknowledging how they perceive the world and so allow or enable it rather than restricting it, that's going to improve your relationship. So that's one way. As a model for humans, I don't think about dog behavior in the same way. Actually, I just study dogs for dogs' sake, right? But I am very interested in what they teach me all the time about observing behavior. I mean, I was trained as an ethologist, a cognitive ethologist, but I'm always learning about dogs' observation from dogs because they're noticing things that I'm not because I've pre-seen it, right? My categories, the human categories, restrict our seeing often, and so dogs open up and remind me of ways of seeing, and also they remind me of ways of smelling, right? As I said, we have perfectly good noses and we don't use this sensory organ nearly as much as we could. I'm kind of surprised by that. So I see them as a model for living more than I do see them as a model for thinking about the human brain.
Yeah, I think both, as I tried to say in the very beginning, both the diversity and the unity are key. Kind of as a neuroscientist, I kind of take the approach of using these fish to try to illuminate general ideas. And because of evolution, if I show you under a microscope neurons of the fish and neurons of a human, in many cases you wouldn't know the difference. The building blocks, the molecules, the way they work are so similar because of evolution that there's, in general, a great deal that we can learn, and we use animals like the fish because for certain neuroscience questions, this happens to be the best model system. So in that sense, the work I do is all geared towards getting the deepest understanding I can of some neural process, and that neural process would be something quite general that would apply all across the animal kingdom. So that sense, yes, but I also agree and enjoy studying diversity for its own sake, and it has value to think about what these animals do and that they have their own individuality and their own lives that have value, irrespective of what they're going to tell us about people.
So on that note, that's all the time we have, but I want to say thank you to all our audience, both here and online, for attending this event. Thank you to our speakers. If you're online, I think there's going to be a survey that's put in the chat, so please take a couple minutes to fill that out and let us know what you thought. And Dr. Lee is now going to close out the event.
Well, first, give everyone a round of applause. Thank you so much to Dr. Horowitz, Dr. Sawtelle, Dr. Zung for this really fun and engaging conversation. I want to thank all of the people who submitted questions and raised your hands and joined us tonight. So I hope you enjoyed your time at this lecture series. If you liked it, I hope to see you at our next one in the new year. It'll be on March 18th, another Tuesday evening, and the topic will be mood disorders. We will have Dr. Stephen Kushner and Dr. Adrienne Jacques Ambrose studying all of these things about how moody we might get and what happens then. So until then, I hope you be well, and I hope to see you in the new year. Thank you.
Thanks.